Good morning to all. So today we see the instrumentation of the IR spectroscopy. What is type of instrument or the different parts of the instrument used to detect or to run the IR spectroscopy. So this IR spectroscopy includes source. First is the source, that is a monochromator, sample holder, detector, and the recorder. While sample handling involves the preparing the solids, that is pellets, molds, and the films. Liquids, that is the thin films in salt plates, and the gaseous cells to ensure transparency in the IR region. Often using the salt plates, that is the sodium chloride or the potassium bromide, and avoiding the water to get clean spectra. Key sample preparation methods for the solids are the KBR pellets, grinding with the KBR, and the usual molds, grinding with the mineral oils. So these are the important parts, important instrumentation parts of the IR spectroscopy. So that is the source, monochromator, sample holder, detector, and the reducer. So these are the more important parts of this spectroscopy instrumentation IR spectroscopy. So first is the source. So this source is emits the IR radiation. So this emits the IR radiations. So the IR spectroscopy starts with this source, emits the IR radiations, which is bombarded on the uh, sample. So sources are the glober or the nurse globe. So these are the two important sources are the used. The next monochromator or the interferometer. So this what is the application of this monochromator? It separates or the encodes the IR light, that is gratings or the FTIR as interferometers. So this monochromator separates the lights or the encode the IR light. Only the it encodes only the IR light. And second is sample compartment. So this sample compartment has application or used to the hold the sample cell or the holder. So this sample compartment it acts as a sample holder, or it is used to keep the sample. Then next is a detector. Sample detectors, what is use of the detector? A detector measures the intensity of transmitted IR light, that is the thermocouples, bolometers, photodetectors. These are the some examples of the detectors. And then a recorder or the computer. So it is attached with the computer. It is displays the final spectrum, transmitters versus the wave number. By using the recorder, we get final graph or the final spectrum of the IR spectrum. So this first is the source. Source emits the lights, and the monochromator it separates the IR region. Sample compartment it holds the sample. Detectors detect the amount of intensity absorbed by the our material. Then a recorder gives the final result. So this there are the some techniques, that is the how. So these sample handling techniques, there are the some techniques by used to the handle these samples. There is the liquid samples are the handle, or the a drop, or the how these sample are the placed in the sample compartment. So first is the liquid, a drop placed between the salt plates. For the liquid, a drop of the sample is placed between the salt plate, that is NaCl or the KBr, to form the thin films. So these thin films are the pumps, and these thin films are the used or the kept in the sample compartment or the sample holder. Then how these solids are the samples are the made. So first is KBr pellet. The grind sample into dry KBr and the press into the transparent pellet. Then usual grain sample with a mineral oil will to form the suspension between the salt and the paints. Then third is a solution. Dissolve in an air transparent solute and place in the solution. The thin films melt and spread solid onto the salt plate. Gaseous samples use a longer path length gas cells made of the IR transparent materials. So these are the some important points also that is must be the transparent to the IR, that is NaCl, KBr, CSIR. Avoid the glass or the plastics. Moisture, if there is moisture is present in our sample. Many salt plate, that is NaCl, KBr are the hygroscopic, so which absorb the water and must be kept to the dry. So these water absorbers are also reflected in our results. So sample purity essential for the accurate identification. If our sample is very pure, so we get accurate results free from the interfering solvents and the atmospheric moisture. If our sample is uh, it with purity impurity, so these impurities of the presence of the water molecules or the presence of the solvent molecules, so this impairing the our results. So our results are the uh, get reflected. So for the getting the accurate results, we get important or the clear results or the uh, very clear IR spectrum. So in the last week, so there are the important parts of these IR spectrometers are the first is source. Source is very important. So first part is the source. Then a monochromator, it is used to the separate this IR regions. Then sample holder, detector, and the recorder.